Hi, everyone, and welcome to today's Build to Sell webinar. And we are happy to welcome Anton Molina to give our talk today. So I will turn it over to you, Anton.
All right. Thank you, Stephanie. So the title of the talk, Nucleus, Building a Synthetic Cell Knowledge Base. And I guess I'll just start with sort of a little bit of a personal introduction for how I sort of got into the synthetic cell field. Um, it, I think, kind of traces back to sort of a little side adventure that happened during my PhD when my advisor one day asked me to essentially build a robot. He thought that would be an important part of my thesis. And at the time, I was a materials scientist, sort of chemistry, wet lab background. And so that was like sort of an intimidating prospect. Um, and and it, led me down this path of like learning about Arduino and learning electronics. And what was kind of surprising was how easy it was, which challenged my intuition. And I got really curious about kind of the history of this object and how it came to be and sort of the design principles around it. Um, and uh, and learn sort of essentially what open source hardware was, right? And so sort of what can be created when you create sort of composable systems with open interfaces that are well documented. One of my favorite images actually is just the very first prototype of an Arduino. So this goes back to maybe 2003 or so, and it's here on the right. He's very primitive, right? There's a handful of circuits, a wire, a resistor, right? And I always find that really inspiring to just see how, that sort of first step was really, really enabling for people because if you don't know about electronics, it's like where do you find the parts? You're working through catalogs and the internet trying to find this stuff. And so the fact that somebody helped kind of package that, put it together, and eventually productize it was really enabling for people, artists, students, undergrads to get involved with this field of electronic, even graduate students like myself. Um, and then and then because of that documentation, open interfaces, right, things grew up around that. And so over here is just kind of like this ecosystem of sensors that came into existence. And companies, right, that would sort of provide the sort of marketplace of sensors built largely on kind of Arduino and sort of the family of electronic components that sort of share these interfaces. Um, and then something even more interesting happened, which is people then put those components together in new and interesting ways. So like early 3D printers were really enabled by, this kind of class of electronics that allowed people to sort of assemble mechanical systems in new ways, right? So here's an early 3D printer, and then, a little while later, right, the combination of easy electronics that are sort of modular and interoperable, along with 3D printed components gives you drones, right? And this is again kind of in the mid aughts, and now flash forward 20 years, right? These are maturing industries that have a tremendous amount of impact in the world, right? So you have like 3D print farms are a thing. There's many companies doing this. Drones are, definitely a thing and are transforming our society. And I find a kind of an analogy in what we imagine synthetic cells can become from this, basic idea of putting together components, modularity, open interfaces, and letting people sort of take ownership and apply these sort of bits to their own problems. And so with that, I'll transition over to the talk and sort of recap a seminar from last month that my colleague Anton Jackson Smith gave, which introduced um, BNext, Nucleus, and sort of the synthetic cell work that we do. So I'll introduce those things, but through a slightly different lens. And we usually motivate this sort of presentation of our work through just collaboration and imagining collaboration at a scale, at a large scale, right? Most biological collaborations are, pairwise, they're small groups, but, mature engineering fields involve collaboration over, hundreds, if not thousands of people building very, very complicated systems. Um, and so kind of our lens on synthetic cells and synthetic biology broadly is this idea of compositional biology, which is just sort of a different framing around the bottom-up lens, right? We work with known components, and then that can be sort of written down, specified, well-defined, and then compose them into, cells and cells of increasing complexity. Taking this approach, we kind of have to 10 steps back since we're starting from scratch. And then the core task is integration, right? People are doing, one module in Germany, another module in the US, putting those together helps us walk up this stack of complexity. And so there's a problem in that, right? Which is that that's still very hard. And so there's sort of this conception that we have that there's an integration gap, right? There's a lot of one-off demonstrations, a lot of individual modules, but they don't kind of work together. And on the other side of that is a really interesting future where the sophistication that bringing those modules together unlocks, can lead to a lot of different, advancements in being able to sort of master biotechnology. But what's missing is kind of the infrastructure and dedicated team needed to do the boring work to help this community put those pieces together. And so, sort of our solution for that is what we call Nucleus. And again, in the talk last month, my colleague introduced Nucleus, and it's sort of this collection of tools that are geared for building synthetic cells. Um, the name kind of comes from this, biological inspiration, right? It's the central and most important part of an object, and kind of forms the basis for its activity and growth. And I think for this community, hopefully this infrastructure and set of tools can kind of fill that role. Um, and in that talk, it was presented as roughly just like a collection of things. And in this talk today, I kind of want to go from a collection and kind of start to paint a picture for how they fit together into something like a coherent workflow that supports this activity of cell building and integration. Um, and just to introduce a little bit of a framework, right? There's sort of a generic idea around just like often called like the research life cycle, which just describes, how a researcher goes from like having an idea to contributing to back to the scientific literature and, kind of growing the knowledge pie for everyone. And so it has these sort of generic components, right? There's a knowledge base, there's a design phase where you conceive your experiment, you build it in materials, you analyze the data as a result of that, and you share it. And today, there's a lot of friction in, well, many different disciplines, but particularly in sort of wet lab biology, to go end to end, right? Our knowledge base right now is, it's kind of locked in PDFs and method sections in sort of guild knowledge a little bit, right? Things people remember what worked, what didn't. We build our systems often, in using non-standard materials, or we're using materials that we don't fully understand because they're, kind of closed formulation and the manufacturer doesn't tell you what's in it. Data analysis is fragmented and often idiosyncratic. And then the way we communicate is also, lacks a lot of context, right? If it's, if I share a graph in a slide deck, it's missing a lot of the sort of detail that makes it valuable for others, or even valuable to yourself later on to remember what you did. And then similarly, kind of the final goal is the, is a publication, which is a PDF. And again, a lot of the important contextual information that's needed for sort of reproducing this and handing this off to somebody else is lost. And so we're sort of missing an opportunity to build a really sort of accumulating knowledge base. And so when we think about, let's see, when we think about, how do we get collaboration at scale? We think a lot about, what does it mean for others to sort of feed into a knowledge base? Also, what should that knowledge base look like? And, what kind of interventions through tooling and materials can we to just reduce the friction and increase the kind of context and like sort of quality of data as it sort of walks along this path of going from, uh, uh, sort of idea to sort of new result, or even just validating existing results, right? That's a lot of the work that we do is just sort of getting up to speed on what's already been done. And so just to kind of structure this talk, I'm just going to essentially walk through each of these steps and some of the components that we've started building and are kind of at this phase now of stitching together and really sort of walking through this workflow internally at BNX, and then also making these tools available for the broader community, and just kind of keep that sort of iterative feedback loop going so that we can continually improve these things. So going back to the knowledge base, we started Nucleus almost 2.5 years ago, back in December of 2023, with basically a documentation page, right? We were basically, it was very painful to kind of parse through just all the different kind of method sections needed to do the key processes that are needed for building synthetic cells, right? Like, piecing together a liposome protocol and realizing that, oh, there's a couple key materials in there that aren't well documented, but are very, very important for making the thing work. I think mineral oil is like one of the sort of famous examples that we often cite around just there's a couple SKUs out there, a couple particular product numbers that work and others that don't, and mineral oil itself is a very ambiguous product. So clearly we needed documentation that included that level of detail. And then, that sort of gave us a foundation to start building on top of that, right? We have different modules. And sort of the properties of them are represented in what we call specifications. Um, and we have different sort of implementations of modules and sort of guides to navigate all this documentation. This is still, these are kind of the course categories for how we bin knowledge, very much a work in progress. But we've also been doing this for a little bit now, and so we're sort of on the second iteration of this in some sense, and making it better and more participatory, right? So down below, you can check out the documentation. We've migrated the site to it was originally a Notion site just because that allowed us to get started very quickly, and we've migrated that to a site that's hosted on GitHub. So if you can interact with that there, post issues, let us know how to improve it, and discussion. We have a forum as well for people to discuss these protocols. Um, Okay, so great. There's a knowledge base. I can, sort of get started on making a synthetic cell, hopefully a little bit faster than having to piece together 10 different protocols, and sort of synthesize them. The next part of the sort of experimental life cycle comes in just defining your experiment, right? And so plate mapping is sort of a key bit of kind of curating this information, and it has a lot of downstream consequences. It's also kind of challenging to do. It's very tedious work. Um, but having a sort of standardized representation, and it can be very simple, right? It can go a long way for, building some of that connective tissue that allows me to analyze this data using kind of a common workflow or hand off that data to somebody else. And so on our docs page, we have some tools that sort of describe how to make a sort of plate map that fits with our other downstream tools that I'll get to in a minute. And there's some sort of convenience functions in there that allow you to sort of define, controls versus reference standards and whatnot. Um, uh, so yeah, so that's uh, that's sort of building the experiment. I think there's a lot of complexity that we can add on to this, and complexity in the sense of making it easier, right? Building a sort of streamlined UI around it. Um, Another part of the experimental process is just materials, right? So Nucleus is sort of a pure centric space. And one of the biggest grievances is that, we don't know what's in the pure system, despite, its best advantage is that it is a defined system. And so we spent a lot of time um, the, building out an open formulation version of Pure that we call cytosol. And the plasmids that we use to make this are, available online. If you're interested in getting them, you can email us. We have protocols that describe kind of how to transform them. Um, and we're on a limited basis, we're sort of shipping out cytosol to a few select groups, but this is another kind of key part of the life cycle that is important for building kind of that sort of Arduino-esque interface, right? It's the physical thing that coordinates everything around it. And related to that on the physical material side is this idea of normalizing standards. I recently just saw this graph. This is from on the left here is two different data from two different plate readers. So the dashed line's plate reader 1, the solid line's plate reader 2, and they're not normalized, right? And so just a couple of colleagues of mine spent quite a bit of time figuring out how to properly kind of normalize across these instruments so that we can make meaningful comparisons between them using fluorescein as a standard, right? And you can see just how if you're not careful about that, you can be incredibly misled about about your data. And so this is kind of a key bit that will help us unlock a little bit of the interoperability that we're after. Um, the next part is analysis, right? So here on the right is a figure from a NIST report that was basically recognizing that everyone in the field uses sort of this de facto assay, the GFP fluorescence assay, but which is great that everyone kind of roughly agrees that that's a thing that should be done. Problem, everyone sort of does it in a different way and leaves a lot of information on the table, right? So it's like what, version, what flavor of GFP do people use? That's, sort of idiosyncratic. How, do we think about endpoints? Do we think about, full kinetic traces? And then if you do a kinetic trace, right, how should you pull information out of that? I think quite a few people leave behind the kinetics for convenience sake. Or if they don't, right, they don't necessarily kind of pull out all the information that can be pulled out because, sometimes the math is complex or maybe, the parameters are slightly different. But, this is the kind of data that, if collected and sort of interoperable, right, can really help build out the sort of digital representation of these systems. Um, and so we sort of took it on ourselves as a starting point for moving towards a little bit of standardization, right, is to sort of build out a toolkit that makes extracting that information relatively straightforward. So this is sort of the idea of the cell development kit, which is sort of our digital collection of kind of software tools that just sort of help in different aspects of these workflows. The kinetics analysis sort of workflow is one example of that. Um, it's available on GitHub. You can pip install it as a Python package. Um, Also, from sort of an accessibility point of view, we make it available on what we call Nucleus Hub, which is basically a kind of shared computer that allows people to access computing environments with pre-installed packages. So, as more and more sort of tools and packages get integrated into this framework, making those available for the community, so you can just kind of log into a URL and access these workflows, access these template notebooks without having to configure a software environment, is a direction towards like making the kind of the right thing, the easy thing by increasing, just the barrier to working with software. Um, and then, this sort of unlocks a couple things, right? I already alluded a little bit to modeling. There's another sort of power that we're prototyping at BNext in our own kind of lab, which is just the ability to make comparisons across many different experiments. I think we started with, okay, the database has to be built with just, using one plate reader because we're still figuring out how to correctly normalize it in a way that we were confident about. But now we're able to kind of take data from multiple plate readers and now build out this sort of plots that allow us to say, hey, these are the materials that we use, these are the conditions that we've run in the past and make comparisons against that. And then over time, right, we have, we're building out kind of the raw data that allows you to imagine informing kind of the pure subsystem models that would help build the sort of digital representation of synthetic cells as we increase the complexity of these systems. Which also kind of provokes an interesting question, right? Oftentimes the experimentalists are sort of separated from modelers, right? And there's a need to be able to transit information between two different sort of geographically separated groups. And that can be kind of tricky and time consuming. And there's a lot of back and forth about, hey, how did this work? What does that sort of annotation mean? And so this is one sort of motivation for why we think also about the sharing part. So we have this sort of tool that we call developer notes. Developer notes are essentially a way of sharing contextualized data. So these are a collection of, you can think of it like a project repository. It includes markdown files, it can include Jupyter notebooks, it can include, some data sets, design files, kind of the whole research project environment, which is kind of shown here in this figure down below, right? And then it's wrapped in that sort of text layer. Um, and and you can sort of bundle that and put it online for others to access. Um, and sort of what one kind of cool and powerful consequence of using sort of this Jupyter-oriented technology stack is that it's very flexible and customizable. And so it's allowed us to work with partners to build out extensions on top of this for highlighting things that we care about as sort of first-class citizens in the presentation of data. So, for instance, here, right, this is a sort of right in the text of the dev note that shows a particular sequence map that was used to collect some data. And so this was a work from a colleague of ours, Charlie Newell, who characterized a promoter collection, and so he was able to kind of show, the full sequence map and show, okay, here's the promoters, and kind of confirming to the person looking at this artifact that the sequences are indeed there and can kind of scrutinize them themselves. Another thing that we've started to embed is microscopy data. So here, right, this is a ZAR formatted image of liposomes expressing GFP over time, and so these can be also sort of embedded from object storage directly into the article, which just provides a greater kind of sense about the data beyond a couple representative images. Um, and then importantly, this is kind of how um, these are sort of, you can think of as developer notes as sort of a staging ground for documentation. So, um, uh, internally we push developer notes to our website. We work with collaborators and for certain work, right? If they want to bring it into the documentation or the knowledge base, right? Um, they can contribute a dev note, and then we can integrate those findings or ideas or particular module into the documentation and just make it better. Um, and so that's kind of what this figure is showing, is that dev notes sort of pipe in. Um, right now, I'm actually at a workshop on sort of thinking about different ways of organizing scientific knowledge, right? So one idea is like a discourse graph, which sort of provides a way of sort of creating simplified representations of scientific knowledge, and putting this idea together of having a flexible format that can be tuned to the needs of the community affords different ways of organizing knowledge that I think can help us just accomplish that goal of collaborating at scale and kind of refocusing kind of the research products and artifacts that are needed for people to work together. And so kind of taken together, right, this points at something that we call like the nucleus development cycle, which is sort of this loop of going from documentation, building and analyzing an experiment using known reagents, and then kind of sharing them back, right? So we can build out this sort of accumulating knowledge base that we call the docs, that hopefully can allow more people to get to the kind of forefront of the field faster rather than reinventing the wheel. Um, and right now we're sort of prototyping this workflow with in a couple different collaborations. So I'm going to call one out here, which is the Schmidt. It's funded by Schmidt Sciences. It's called the Developer Cells Collaboration, and um, and so we're getting to the point where some of the participants are using these tools to kind of help you know uh, uh, document their work along the way. It's also really important for us, right? Because we're sort of acting as a validation and verification support partner. And so in order for us to kind of keep track and play that role, right, we need to be contextualized in the work, and having this sort of very data rich sort of representation is helpful for us. Um, and and then one thing kind of comes up, BNX is a company, Nucleus is this sort of collaborative, open source-esque project. It is open source. It's things are licensed under sort of CERN, using CERN open hardware license and sort of Creative Commons licensing. And it's like, how does BNX relate to that? And I think it's sort of well exemplified in this collaboration where um, uh, the researchers are building on the open sort of tools and resources, and then BNX is sort of providing materials in the form of cytosol, training and sort of coordination in the form of workshops, and also doing kind of that verification of the kind of final research projects. So there's sort of an R&D angle there. Um, uh, and then, another question that often comes up is just sort of how to get involved. And we've been sort of incubating this project for a while now, and have, it's getting to a point where we want more and more kind of participation at all levels. And so here's just a kind of a couple paths, right? If you want to make a technical contribution to Nucleus, invite you to email email me about, kind of here's what I have in mind. A lot of people are like, have kind of published something already and just want to make their thing more accessible to others and have it be documented. Um, so DevNotes is a great vehicle to do that, and happy to chat with anyone that's interested. GitHub is another sort of interface, right? If you're reading the documentations and say, hey, there's a typo there, or this doesn't quite make sense, you can write an issue. It's super easy. You just sort of, type something up, write a comment, we'll pay attention to it. There's also the forum, which is sort of a just general discussion space that we're trying to create, and create a kind of space for people that are just interested in sort of engaging with the whole collection of things that we think about. Um, and then another kind of way of participating um, is we've been doing this sort of canvassing of the community. So last year we initiated a survey of the synthetic cell community through an independent third party called Conscience, and wanted to just really get a sense for what is the state of collaboration in this field, and where do people want to take it? What are they concerned about? What are the different sort of challenges and opportunities? Um, and so that's online now. You can read it. We recently kind of finished a sort of did we hear right kind of survey, just to make sure, just to kind of open it up and make sure that we got kind of a really good feel for the sort of zeitgeist. And we'll post the results of that soon. And so we're now kind of moving into the second phase of starting to address just how. So I described kind of stage one. Now we're in stage two. Stage three is, I think, kind of moving towards just how do we, it's about building collaborative structures, right? And everyone kind of has a little bit of ownership in this if they've contributed. So how do we manage that? So we've got kind of want to move towards providing more concrete answers for that kind of in towards the end of the year. Um, for the next kind of month or two, I think we're going to kind of put out some writing that sort of describes some of the questions that we've gotten a lot about Nucleus. Just like I alluded to earlier in the talk, what is the relationship between BNX and Nucleus? Our last Build a Cell seminar in June, where there are a lot of questions around sort of how does IP work? What are kind of the details and things to consider there? We have some thoughts. They're not well articulated, and so we want to kind of share our thoughts and invite discussion. Another priority is a roadmap. If you've been following our work, we've at intervals sort of release sort of point updates, right? And it's sort of V2, V 0.2, 0.3, 0.4. We're now at, I think, 0.5. Eventually we want to get to sort of this milestone of a V1. What does a V1 sort of infrastructure and collaboration structure look like for this community that can sort of scale and be, really sort of participatory and just, functional, and help upgrade this community in terms of what we can accomplish together. And then, another kind of topic is how to, sort of manage and sustain and govern what we build together. And so with that, I guess I'll say, funder acknowledgement, shout out to the team at Be Next. Let's stay in touch. Feel free to reach out. My email's right there on the side. And there's some link outs to our websites and the conscious report. And so I guess with that, we can take questions.
Great. Thanks, Anton. That was uh, great to hear. If people have questions, feel free to unmute yourself or put it in the chat. Right now, I don't see any questions. So I guess to start off, how are you? So it's the Sincel community, but are
Mhm.
there other collaborators that you've reached out to, or how are you advertising to potential folks?
Yeah. Conferences. So we've done a lot at sort of Sincel. We'll be at Seed and IWBDA next month, and then sort of a waterfall. list. So kind of going down our mailing list of people that have signed up and people that have sort of signaled some engagement in the project already. Um, I think kind of the next step is sort of the adjacent sort of communities that are sort of at the edge, and we haven't quite broached into those communities yet, at least not in a sort of intentional way.
Got it. And I realize you said that you're kind of at the beginnings of figuring out a more fully fully developed idea about IP and how that works. But
Hmm.
and there were a lot of questions in the past seminar. But if
Yeah.
people want to contribute, I guess how does open source and IP work with their data?
Yeah. So there's a couple layers to that answer. So, in general, IP is not a significant concern, right? In the sense of, there's patents and stuff like that. That's usually kind of where the discussion gets challenging. Our general policy on like dev notes is that it's just, CC BY. So, it's pretty consistent with most, kind of the default for preprint stuff, right? You own the data, you get attribution, and people are free to use and modify as needed or as they want. And then others overall, the project is held with or sort of falls under a CERN open hardware license, which kind of affords a little bit more specifics around kind of the formulation and sort of patentable things. Um, and so so if things are patented, then that becomes a complicated conversation. If not, then it's a really sort of clear indication that this is something that can be used by sort of others outside of the academy, which gives it, kind of a big impact in the world, right? Sort of like Arduino, right, is something that can be incorporated into commercial products if needed, and sort of unlocks that sort of ecosystem of different real world applications.
All right. Do people have any questions? Like I said, feel free to put it in the chat or unmute yourself. We'll wait a couple of seconds.
Mhm.
Have there been any surprising communities you reached out to? So obviously you're well involved with Build to Sell, but have there been any other ones that you've started some collaborations with?
Yeah, I think this one's still sort of baking, but sort of in the protein design world, I think there's a lot of sort of interest that we've had, and I guess one of the challenges is while we're still sort of building out these workflows and just how things kind of stitch together, there's still a lot of friction. We have limited bandwidth, right? And we're still have to balance our time. So getting kind of new people on board is a slow process. But the protein design community, I think, is a really just exciting sort of natural connection between synthetic cells and sort of their world. So that'll be a new frontier. The other natural one is sort of the kind of biophysicists. I've had quite a few conversations with people that just kind of think about sort of what might be afforded through having kind of the spherical cell or spherical cow for cells. Um, so I think those would be sort of the two frontiers to kind of bring in through this framework.
All right. And all right, last call. Does anyone have any questions? All right. Well, I think that's it. Thank you, Anton. This was great. And we will be posting this video shortly. Thanks to everyone. Have a great afternoon.
Thanks. Bye,
Yep.
Stephanie.
Bye.